I think Shinyu's involved in the project, and Lee. Good. Well, thank you very much, everyone, for joining us. And just to be aware, there'll be a few more people joining us as we start. So I'd like to start off with welcoming you all. And the very first and important thing to do at the start, of course, is that we acknowledge that we are on the lands of the Wurundjeri people. I'm actually on the lands of the Bunurong people, where I am now. And these people who have been custodians of this land for thousands of years, and we very importantly acknowledge and pay our respects to their elders past and present. So thanks very much. So this, we're very fortunate today to have a seminar. Our university project, which is funded from Chancellery Hallmark Research Initiative, which is on future foods, focuses on the production of alternative proteins, including from microalgae, plant-based yogurts, through fermentation, blended plant and animal products, as well as cultivated meat. So we not only cover technological, nutritional and health aspects and consumer aspects, but also regulation. So please see the website in the chat if you're interested to find out more about this project. Hence, on the, I guess on the technological and development sides and even the regulation side, this presentation today by Associate Professor Anna Dilger is very relevant. And importantly, I think the lead into this presentation by Anna is that in 2020, CAST, the Council for Agricultural Science and Technology in the USA, they published a commentary called Producing Food from Cultured Animal Tissues. And in the link for the seminar, there's actually a link to this. There's actually a link to this publication. And Anna was a key person involved in this. And I also saw a very good presentation by Anna on this topic. And so this outlines the state of the science of cultured meat. And today Anna's going to give us a report on this, as well as some updates. So we're very fortunate to have Professor Anna Dilger. She's a professor in the Department of Animal Sciences at University of Illinois. She specialises in meat science and muscle biology, and she has degrees from both Purdue University and University of Illinois. So her research actually focuses on the growth and development of livestock, in particular pigs, and how maybe changes in diet management and genetics can influence meat yield and also quality. At Illinois, she certainly does some teaching at both undergraduate and postgraduate level, and especially not only on animal issues and meat production, but also on animal growth and development. So she's currently the president for CAST, and involved in their task force liaison for their commentary about cultured meat. So she's coming to us with a lot of expertise. She's not only involved in this, but she's also a very good teacher. She's actually been ranked on the university's list of teachers ranked as excellent, and she's received recently an award for Outstanding Young Teacher Award from the American Society of Animal Science. She's a member of several very good journals, Journal of Animal Science and Meat Science and Muscle Biology. And importantly, Anna's, I think, really got this expertise for outreach and communicating scientific discoveries to the public through her involvement in those journals, as well as another one called Animal Frontiers. So I'm going to hand over to you, Anna, to give us your presentation. Thank you very much.
Thank you, Robin, and thank you, Min, for the invitation. I am coming to you from Thursday evening in Illinois, and so I have warned my children that I am giving a talk, and so hopefully they do not come and visit me. But I think it's nice always to give the disclaimer that we might have a visitor, as from time to time we are now doing these things from our homes. And so I really appreciate the invitation. And what I'm going to bring to you, this evening, you're this afternoon, is a report that the Council for Agriculture, Science and Technology put together, really not trying to answer all of the questions about the ideas of producing food from cultured animal tissues. Our goal really was to raise the questions that are out there and bring to the forefront the questions that need to be answered as these products are coming to market. And so In case you are not familiar with CAST, the Council for Agriculture, Science, and Technology, we are actually celebrating our 50th birthday. So founded in 1972, our goal is to provide balanced, credible, science-based information about food and agriculture. And our main audience really is policy and decision makers. And so we do that by bringing together experts to write commentaries and issue papers. And I invite you all to take a look at our website. We cover the gamut of all agriculture issues, being plant, animal, food, environment. And what I like about the papers that we put together is that they don't try to convince you of things. We really are interested in presenting the consensus of science. And as you all know, sometimes the consensus of science is we don't know yet. We need more information. We need to do more research. And so you can connect with us in various ways on social media and through our website. And I am happy also to share these slides after the presentation if that's useful for anyone. And so as Robin said, in 2020, and so here in the US, very early in kind of our COVID lockdown, we presented this commentary called Producing Food Products from Cultured Animal Tissues. And I think it's important to acknowledge that I am just one member of this team. And so you can see here the various authors that we had involved directly in the technologies of cultivated meats. That would be Min-Ho Kim and Jess Krieger. We had some meat scientists and also an attorney, because a lot of what we're talking about here really has to do with regulation and how these products will be regulated. And so I do absolutely want to acknowledge the work of all these wonderful authors and our reviewers that put together what I think is a really strong commentary, really to raise these questions about this kind of new technology. And so that was our goal, was identifying the challenges and the uncertainties regarding what we all believe is eventual products that are going to come to market that are produced through the culturing or cultivation of animal cells. And so the questions that we centered around on this report were informing policymakers about, what are we talking about? What are the techniques that are going to be used to make these products? And what advances in technologies are needed before we start seeing these products really in a widespread way? What are we talking about? What are the types of products that we expect to come to market? The big question everyone always asks is when? Right? So at this point in time, there is one product that has been produced on market. And I think there's a lot of companies that are close. And so we wanted to kind of put some timing on that. And then probably from a policy standpoint, I think the stickiest issues right now are around who is going to be responsible for regulating these products and how is that regulation going to take place. And so what I'm bringing to you today is a very US focused approach for how those products are going to be regulated. And I'm hopeful at the end of the presentation, I would love to hear how governments around the world, particularly your government in Australia, is starting to approach these products from a regulatory standpoint. When we talk about regulation, it really comes down to how products are labeled. And then last, we're going to talk just a little bit about some food safety risks. And so for this particular audience, as you have this project focusing on the future of food, for all of you, this particular question probably isn't the one that you need answered, right? What techniques will be used to make these products? But for our commentary and for communicating with the public, I think it's really important that we develop a strong and simple story of how exactly we're talking about making these products. And so at CAST, we broke this down into kind of a 4-step process, developing and identifying the cells that are going to be grown, growing those cells, or what we call cultivation of those cells. And then maybe a little bit further out there in terms of where we are in technology is the idea of tissue structuring and cultivating not just cells, but cultivating entire tissues. And so from a cell line development standpoint or a cell line selection standpoint, there are, of course, a lot of potential sources of animal cells. And the different companies and research labs that are involved in cultivating cells now have made use of all these different ways to obtain cells to start their cultivation process, whether that's biopsies from living animals, the use of embryonic stem cells or immortalized cell lines, or bone marrow and other inducible stem cells. Right now, my survey of what's currently going on is that mainly those cell targets are muscles and fat cells, adipocytes. And that makes sense, as that kind of makes up the bulk of the main cell types that we have in conventional meat. But you can absolutely understand that as we talk about moving from just cells into tissues, there are lots of other cell types that we're going to need to develop cell lines and select those cell lines if we expect to have blood vessels, connective tissue, nervous tissue alongside our muscle and adipocytes. And so when we think about cell line selection, we're not just talking about what I think is challenging is you actually have to select for a couple of different things. It's not just the fact that you need cells that grow, divide, and mature in culture. And selecting for those types of cells kind of is a suite of traits that's a lot to do with growth. What we don't know is how exactly to select cells based on, I'm going to call them more meat-related things, nutrition, flavor, texture. And I think as a meat scientist, there's just so much that we don't know about things like meat flavor that I'm not sure that we are in a good position to tell somebody, oh yes, for the best tasting cultivated chicken, you should use a cell from where and what chicken. And so I think that alongside this idea of selecting cells that perform well in culture, I think there's a lot to be learned about selecting cells that then perform well in products. And so once those cells have been selected, the next step is to grow a bunch of them. And so we have to cultivate these cells at a large scale. And so as you know, it's right, culturing cells is really not a new thing. It's a technique that we've been using in research labs for decades. What is a new thing is the idea of moving from these really well-established benchtop techniques that take place in milliliters or maybe a few liters at a time, and scaling that up to thousands or tens of thousand liters in bioreactors. And one of the big challenges in doing that is thinking about the volume of cell culture media that will be needed in order to feed those cells. And then thinking about what happens to that media once we're done with it, right? It becomes a waste stream that needs to be dealt with because that media is not necessarily going to be incorporated into the food product. We also know that many of the companies that are developing these products have also been developing new medias that are animal-free, right? From a traditional cell culturing standpoint, we think a lot about fetal bovine serum, right? That is kind of that magic mixture that we would use to have all of the growth factors needed to direct the work of those cells. But for the goals of many of these companies, using fetal bovine serum doesn't fit with their kind of overall company goals. And so developing animal-free mediums, antibiotic-free mediums, there's been a lot of work that has taken place and probably still needs to take place about developing these medias to be used at a large scale. And so cultivation of cells and bioreactors is really the potential first step, and is likely to be what we see first come to the market, is cultivated cells that are then made into meat products. But when we think about kind of the holy grail of this kind of idea of cultivated meat, people don't just want to eat cells. They want to eat steaks, and they want to eat roasts. They want to eat things that look like conventional meat products. And those are not just cells, those are tissues. And complex tissues that put together many different cell types in a particular arrangement. And so in order to completely mimic the texture, taste, and composition of traditional meat, we're going to need to move past cell cultivation into tissue structuring and tissue cultivation, where cells can be seeded into matrices, either of plant or animal origin, and then cultivated as a tissue and grown as a tissue. And so honestly, this is going to require the development of technologies that don't exist today, and the refinement of technologies that do exist today to really accomplish this goal at a large scale. What I find really interesting is if you have encountered people who are involved in the kind of the research at a university level about the idea of cultured meat or cultivated meat. At least in the US, it's very common that those labs have maybe one or two graduate students or postdoc that are working on cultivated meat, and everyone else in that lab is actually working on biomedical tissue structure, such that what they're interested in is growing new hearts for people or new livers for people. And then I've always wondered whether it's a little bit strange that there's these two graduate students over there that are eating their experiments. And to me, it always seemed like kind of a weird mix in a lab. And so when we think about what has to be overcome before we're going to see not just one product on the market, but dozen products on the market, and products on the market at a cost which is competitive with both traditional meat or alternative kind of plant-based meat substitutes. And so in our commentary, we identified these technical challenges. The selection and characterization of those cell lines, lowering manufacturing cost, and that includes both media components and also the cost to operate those bioreactors, scaling up. It's not, producing one hamburger doesn't do you any good. You need to produce thousands of them. How to deal with the waste streams, spent media, whether it could be recycled or reused. And then I think right now, one of the biggest challenges is the development of allied industry, like media production, but also the development of a skilled workforce. This is a different workforce than traditional meat production. And to a certain extent, it's a different workforce than current bioreactor production that's making kind of biologicals. And so really, this is what we identified as the targets for the challenges to be overcome in order for this particular type of product to succeed. And so when we think about what are these products going to be, we classified them into 3 phases based on how quickly we think these products are coming to market. And those phase 1 products are really a mix of cultivated cells and meat analogs. What we would consider right now are alternative proteins, our plant-based meats. And then those animal cells would be mixed into these plant-based products in order to provide nutrients or to provide flavor to the overall product. And so some of the kind of anecdotal things that I have heard is it might take maybe 5 to 10% animal cells in a plant-based product really to impart the flavor of beef, which again, I think is really interesting because I'm not sure that we have a really good handle on exactly what beef flavor is. As we move from phase 1 to phase 2, we expect that products are going to be composed entirely of cultivated cells. And they're going to take on a structure that looks like current formed or ground products like hamburgers or sausages or nuggets. Before much later, we expect to see those phase 3 products that really are those cultivated tissues that mimic our current whole muscle cuts. Of course, one of the things that has changed between when I first gave this presentation in April of 2020 and today is that we actually have seen a product come to market and be offered to consumers. And so in December of 2020 in Singapore, they approved a cultivated meat product that was a chicken nugget. It was produced by the company Eat Just and it was a very limited release. And so it was sold in a single restaurant, really in a sample, like a trio of sample dishes. I think one was like in a little bun. One was a chicken on a waffle that you see here. I don't know if in Australia chicken waffles is a thing. It's a big thing in the US, chicken waffles. And so right now we're honestly kind of sitting in anticipation of more products coming to the market. I think for a lot of the companies, it makes some sense that they were slowed by the pandemic. There were a lot of companies that went into complete lockdown and had to pause production and development for quite a while. But Upside Foods, which is formerly Memphis Meats, has gone public with that they expect to have a product in the market this year. And so it will be interesting to see as these products come out, maybe what kind of differentiation that we have. And so as I said at the top, one of the stickiest issues really for a new product coming to market really is the regulation of it. And in our commentary and what I'm bringing you to tonight really is a very US focused thing. And so first of all, my apologies for that, but this gives you an example of at least how the United States is approaching the regulation of that. And so in the US, we actually have 2 federal agencies that regulate the safety of food. We have the Food and Drug Administration, or the FDA, and we have the US Department of Agriculture, or the USDA. And you can see here the current areas of responsibilities of the FDA and the USDA with respect to animal products. And if you're looking at these lists going, wow, that's a really complicated way that you all have decided to regulate things. Yes, agreed, we agree. Such that primarily meat is regulated by the USDA, except for when meat is mixed into other products, when it's regulated by the FDA. And when we think about things like veterinary drugs and devices, livestock feed additives, and biologicals that might be used in animal production, those are also regulated by the FDA. And so it wasn't surprising that as these conversations started kind of in earnest in 2017, 2018, both the FDA and the USDA felt that they had jurisdiction, they had expertise to offer in the regulation of these products. And so they have decided to have joint regulation or joint jurisdiction over these products because they feel that these cultivated cell products really span the areas of the FDA and the USDA. And so in 2019, they signed a memorandum of understanding outlining their planned cooperative approach. And this is how my simple brain understands it. When cells are cells, and they are in bioreactors, they're being selected and cell line developed, that's going to be the responsibility of the FDA. And honestly, that makes a lot of sense. That is where FDA's responsibilities and kind of expertise already are. That's very similar to the production of biologicals that the FDA already oversees. However, when those cells or those tissues leave the bioreactors and really become something that's going to be manufactured into a food product, that jurisdiction is going to switch then to the USDA. And so at that point in time, it's going to be treated like meat, the same way that the USDA would treat all other meat products. And in particular, because the USDA is going to assume responsibility for regulation and oversight of that final product, it is the expectation that those products are going to be subject to the same regulation as conventional meat. And so in the US, that means the requirement of HACCP plans and sanitation protocols, the inspection of products and records, the presence of inspectors at all times when meat is being manufactured, and the pre-approval of labeling. And it really is kind of that labeling issue that's probably created the most controversy or the most discussion in the United States. And so the USDA has been charged with developing any additional requirements that would be needed to ensure the safety and accuracy of labeling of these foods. That really, like, everyone wants to know, well, how are these products going to be labeled? And actually in my inbox this morning was a podcast that was like, update to labeling. And I thought, oh no, something has happened. I'll have to update my slides. No, we still don't know. And so it's really a big area of uncertainty about how these products are going to be labeled, because so far in the US, no product exists to be labeled. And so what is needed is a company to submit to the USDA a product and their proposed labeling for the USDA then to approve it. And so far, no one has done that. But it is assumed that the USDA will apply the same kind of format that they currently apply, and that's through what we call standards of identity. And so all meat products in the US are identified by standards of identity, kind of a really complex definition of what makes a hamburger or what makes a ham. And so the question is how the USDA will be able to apply these definitions that already exist to these cultivated meat products, or whether or not they're going to have to expand those definitions or make new definitions to really encompass these products. And so what I have here are just 3 example standards of identity to kind of pose some questions that I don't have the answers to. And so when we think about something like hamburger is chopped, fresh, or frozen. It's beef. It contains both beef and beef fat. In the US, it can't contain more than 30% fat, and it can't contain things like water phosphate binders or extenders. When we think about the formation of those phase 1 products that are a mixture of animal cells and kind of plant cells, we would expect them to have things like binders. And so under the standards of identity, they could not be called hamburger. And in order to be hamburger, would they have to be cultivated cells from beef animals? Would we have to label the source of those beef cells came from an animal versus cultivation? When we think about something like ham is defined as coming from the pig leg. And so if you were going to make a cultivated meat ham, were you going to have to cultivate pig leg cells? I don't know. How specific are things going to be? And will companies want to have something that is a cultivated ham? And so we kind of exist right now in this weird unknown because we don't have any products to label yet or proposals for the USDA to react to. But one thing that was noted on this podcast I referred to this morning is, at least from a US perspective, if the federal government, if the USDA issues a label, then that is the label for the country. And it is not then up to individual states. And so right now in various state legislatures, especially when we think about our states that are perhaps heavily invested in beef production or pork production, there is discussion about, in our state, we don't think it should be labeled that. That gets really confusing when you have different labels in different states. And so I think most people are hopeful that the USDA will make a labeling decision, and we don't have kind of a weird state by state type of thing. And so the reason that labeling is I think so sticky and so difficult is there are lots of different names that have been proposed for these food products produced from animal cells and culture. For some people, they call it cultured meat. Sometimes we call it cultivated meat, clean meat, in vitro meat, tissue engineered meat, synthetic meat. I asked recently a class to come up with like, what would you call it? And I had 200 students in that class, and I think they came up with 300 different names. And so I'm not even sure as meat scientists, like we haven't decided what to call it. And so in particular, I like this survey data from 2018. And so it was from Consumer Reports, which is kind of a national survey organization here in the US. And they asked people a question of, if you were going to purchase a product like this in the grocery store that's made from animal cells and looks and tastes like meats, how do you think it should be labeled? And it was interesting to me that kind of people split half and half. Half people said that, well, that should be meat, but like with an asterisk, right? It's meat, but an explanation of how it's produced. And half of people are like, I don't think that's meat. And then there's 5% of people who said, well, sounds like meat to me, so just call it meat. So they then took that a step further and said, okay, so if we're going to call it meat, but with an explanation, what name would you call it? And people were allowed to pick up to 3 different names. And you can see here that I'm not sure that there was a winner. Lab-grown meat or artificial and synthetic meat seem to resonate with a lot of people. But if you think about from a company perspective, from a marketing perspective, I'm not sure that that really is what is going to trip people's trigger in order to purchase it. And so I think that even as a meat industry, we haven't settled on what we're going to call it. And so taking it the next step further about what people, everyday consumers that, let's be honest, don't have a great understanding of how we're producing meat today, how they're going to approach something that is now produced in a very different way, I think is going to be a huge challenge, and something that I think we need to step into that gap and have conversations with people as these products come to market. And so the last question that our commentary addressed had to do with food safety. Because anytime a new product comes to market to be consumed by people, it must be evaluated for potential food safety risk, be those biological, chemical, or physical hazards to food safety. And so when we think about these products, again, because they are not available in wide-scale production, it's a little bit of a black box to determine what exactly those food safety risks are going to be. But we can expect that they're going to be a mix of familiar threats that we know of, that are threats to meat safety, even in our conventional products, but potentially novel ones. When we think about growing these cells in bioreactors, the components of the media, how exactly that harvest process happens and what spoilage might occur during that process. And so there's a lot of claims right now that one of the reasons that we should embrace cultivated cell products is because they're going to be safer than traditional meat. And some have claimed that the lack of the slaughter environment will eliminate the food safety risk that we typically worry about when we think about conventional meat production. And because these will be cultivated cells and not slaughtered animals, that we won't have to worry about food safety risks. But you don't have to be very much of a meat scientist to realize that while the slaughter environment definitely is one concern when we talk about food safety risk, we would expect that any food product that contained animal cells would be subject to potential contamination in that manufacturing environment by kind of environmental organisms that are present in the processing and packaging environment. And so what we did is try to maybe put together a framework to think about these different food safety risks. And so when we think about, okay, so we have cell lines and we're cultivating these cell lines and selecting these cell lines, One of the things that is very novel about that idea is the contamination of cells during collection or the contamination of cell banks with cells that we didn't intend, be those yeast, bacteria, or simply not the animal that we were interested in cultivating. And so that, to a certain extent, is perhaps a little bit analogous to standards of identity, but it's applied in a very different way. When we think about food safety risk in the cultivation of those cells, there are going to be reagents used in that cultivation that currently don't have what we would consider GRAS status or generally recognized as safe. They have been developed for use in labs and are not necessarily, haven't gone through all of the testing to determine that they are food safe. And we would worry about them potentially leaving unsafe residues in products. But to a certain extent, that risk might be similar to the risk that we worry about different veterinary products or food additives in traditional meat production. And so maybe it's possible that we could apply those same type of regulatory principles to these reagents that we currently do to veterinary products or feed additives. When we're cultivating those cells, absolutely, we would worry about contamination with bioreactors. If anyone has ever cultured cells in your lab before, you know that yeast and bacteria are always a threat to contaminating your culture. And so a little bit, that's similar to maybe the risk of animal disease when we think about our traditional meat production. But then when we move to kind of that post-harvest, we take those cells out of those bioreactors and we use them to manufacture meat. Those actually, I think, are the food safety risks that we have the most familiarity with, because we assume that they're going to be analogous to the concerns that we have in the processing and packaging environment in our traditional meat production. And so when we think about, right, the importance of sanitation protocols, the importance of HACCP in identifying where our critical control points are such that we can make sure that we are producing safe products. And so, as I said, that cast commentary is available to you. My goal with speaking with you tonight really was to bubble up these particular questions that we had and to open a discussion with all of you. I am by no means like a world-renowned expert in this area. I think like most of you, just find it really interesting and really fascinating. And so I'm happy to take any questions you have. I would love to hear about your projects and how you are approaching this idea of cultivated meat products or cultivated animal cells being incorporated into food. Because I think it used to be something that we consider to be like out there in science fiction. And I think it's something that we're all going to have the opportunity to eat very, very soon. And so I appreciate the invitation and I look forward to the discussion.
Great, thanks, Anna. That was a really, really interesting presentation. A clap coming through from Gordon. We've already got a question coming in. So on behalf of all of us, I'd like to say thank you. Really interesting, lots of food for thought. And I just said in the chat, please, Brooke's already raised her hand, which is great. So you can either type your question into the chat function or raise your hand and ask it live. So I'll hand over to Brooke's involved in our work, in fact, Anna, just so you know. Brooke Ferrugia. Thanks, Brooke.
Thank you. And that was a fantastic presentation. I am new to the space of cultivated meat because my background is biomaterials, tissue engineering and extracellular matrix, not for muscles, but for other tissues. And it was like, it's not that I didn't know it, but it's just also fascinating to see, as you touched upon, the parallels between the regulations of meat, the regulations of, I guess, biologics and tissues and all of that sort of stuff. The one question I wanted to ask, and apologies if you did touch it, and it's around also, I guess, the public perception of this. Has it ever been, I guess, asked about, because if we're using cells, and obviously it's going to depend on the source that they come from, but if we're getting a cell that's going to be able to be used, I guess, in a large scale, it's probably going to be genetically modified. And I guess, has that question been brought up to the public in terms of, because I know for other types of foods, I guess the public perception of GMO and I guess how that affects sorts of things, has that been brought up and I guess discussed in a public sort of forum?
So I think it's a really, really excellent point. I am not aware of like any survey data or that type of thing that would really speak specifically to the concept of, are consumers who are excited about cultivated meat products willing to consume genetically modified cells, or do they expect those cells not to be genetically modified? And if you look at the various companies, and there are dozens of these companies that they're developing these products. Some of them have been very, very public in that they will not use genetic modification. And others are a little bit not as public about that. I think you bring up an excellent point that if we're talking about optimizing cell growth and culture, like genetic modification is the easiest thing to do in order to do that, right? To remove those the different genes that we know limit growth in culture. Like, let's get those out of the way. And so the question really is whether is who the consumer is for this product. And my impressions of it is there's not a single consumer of this product. I've had, so like I alluded to, I teach a contemporary issue course in the spring to our freshmen. And so I had 200 like 18-year-olds. And it was really interesting to ask them, like, are you going to eat this? And like about 20% of them were vegetarians, and they were split half and half about whether or not they would eat it or not eat it. There's been questions around whether or not these products would be considered kosher. Would they be considered halal? And right, part of the problem, Brooke, is like no one knows the answer to any of that. I think it's really great questions, but I think we will, right now we're talking about this product as if it's one product, right? It's a monolith, right? And it's not. We're going to have a differentiation of products. And I think some of them might be genetically modified and some of them might not be. And some of them might use animal-free media and some of them might not. And some of them might be kosher. And some of them might be more acceptable to vegetarians. And so, yeah, we're kind of in that. Part of why I like talking about this is like no one knows the answers to them. And so you can just ask really good questions.
No, thank you.
Yeah, thanks. Great answer. And I'm going to hand over now to Gordon Lynch, who's with our Centre for Muscle Research. There's a lot of research on muscle and is involved in our work in the project with cultivated meat. Thanks, Gordon.
Well, thank you, and a really interesting presentation. I thought the US was a little bit more advanced in the regulatory side of things, but it's still an emerging sort of field. And I guess my question relates to the influence of the Europeans, because they seem to be a little bit more advanced into the origins of these things, or especially from an industrial scale perspective. The Europeans have been at this for a bit longer. Is the US regulatory bodies influenced by what's happening in Europe or has happened in Europe, or is it just going separately and establishing their own?
And so it's a really good question. And I think that the US approach and the European approach are maybe just a little bit different. And some of it is the concept that the US has a complex regulatory environment, right? I gave you an abbreviated division of responsibilities between the FDA and the USDA. And what we did not dive into are all of the sub-organizations within all of those organizations. And so my impression is that they've been, the US regulators are maybe not the most proactive. We have a regulatory system that's a little bit more reactive, such that at any time, a company could come forth and say, we are going to produce this product, and this is the label that we would like to put on it. And at that point in time, then the USDA will go, yes, we agree. But until that's put out there, the USDA is not going to come out and say, this is the product, this is the label. And so does that make sense from a more of a reactive than a proactive type of regulatory stance is kind of where we're at. And my impression is that other jurisdictions, other governments have decided to take more of a proactive stance in kind of getting out there before the products are available to say, we know this is coming and this is kind of what it's going to look like. And that's just not kind of the regulatory stance that the US takes.
And if I can just follow up just briefly, thinking about the sorts of approaches and the degrees of complexity, because there can be very simple mixtures of different types of cells to produce flavoursome, nutritious products compared with things that mimic steak or seafood. And I just wonder how are the regulations, and is it easier for just getting the nuggets, etc., or burger mince approved, or when we start getting into complexity, does the regulatory complexity increase?
It's not something that I have considered, right? To think about whether or not from a right, I think that when we start thinking about those phase 1 products that are going to be 90% plant or plant fungus, right? Pick your matrix with a very small amount of animal cells. I am not sure that anyone is asking for that to be called meat. And so from not necessarily from a regulatory standpoint, but from a public perception standpoint and from a controversy standpoint, I think those phase 1 products are going to be an easier lift. The closer we get to something that if you set it in front of a meat scientist and said, where did this come from? The closer we get to a product that perfectly mimics a steak, I think that yes, it does get more complicated. Because then you're starting to, it's not just, I can't look at that product, I can't do tests on that product to identify what it is. That starts becoming kind of a regulatory chain of where it came from. And so more of a process verified type of label. Luckily for us, I think we are a long time away from that. I really do think that the concept of engineering an entire tissue, first it's going to happen, at least hopefully first it's going to happen to replace people's hearts and kidneys before we really start thinking about eating them. That's my bias and not a cast position. That's me. And so I think we have some time before we have to start approaching that issue. But I think you're right. I think it does get more complex as we get closer and closer to something that looks exactly like meat.
Thank you very much.
Thanks, Anna. And I'm going to hand over to Hope Johnson now, who's also on our Future Food Hallmark, and she's on from the Faculty of Business and Law at, trying to remember now, Queensland University of Technology. So Hope was, I think, going to maybe even address where we're up to with regulation in Australia. Is that right, Hope? We got a question as well.
Yeah, I'll be fast. Thanks so much, Anna. It's so great to hear a talk on the regulation of these things, because otherwise I feel like I'm the only person looking at this. So it's nice for that. And I've looked at the US and published some research as well on the US regulation in this space. But broadly in Australia, we're planning on taking the same approach that we take to novel food. So we have a proactive regulatory regime where we do a safety assessment that then it'll have 2 different public submission inquiry parts. There'll be a regulator that will consider both the safety of the product, mostly reliant on industry evidence, and then also the submissions that it's received, and will decide if it will allow it, and if so, under what conditions. And we've recently had the Impossible Burger was approved in Australia, and a real focus of the submissions there was on the GM component. So probably that will be a contentious thing. We've also had a big Senate inquiry here into definitions of meat. And they said that they're not really going to consider cell culture at this time, but that they expect it to be something that FSANZ, that's our food regulator, will consider as part of the normal regulatory framework. I don't know if that will end up happening, because we, like the states in the US that have the definitions of meat legislation, have a strong agricultural lobbying group. So maybe we will see a specific regulatory regime be created that is for the approval of these products, or maybe it will just continue through the traditional avenues that we have set up. I'm not sure about that. But yeah, it's certainly been, and it's an interesting area. So thank you very much for that.
Yeah, and I think, right, I can absolutely imagine that in Australia, if I think about the very strong and robust animal agriculture lobbies in Australia, very similar here in the US, that there is definitely some positions of protectionist, right? That if you are a beef producer, you feel pretty suspect about somebody calling that beef, because you feel pretty personal about the product that you're producing. What I have found really interesting from a meat science and meat company standpoint is really the scientific embrace and the company embrace of these ideas of these cultivated proteins, right? These cultivated cells being another source of protein. And so I'm not sure that the meat industry and meat scientists see it as much of a threat as really kind of those animal agriculture, those ranchers and farmers really are the ones that are perceiving that threat. But it definitely has led to some, I'm going to call them lively discussions, some raised voices, and right. And it's hard when I think some of the companies who are out there developing these cultivated meat products are doing so in a way that's a direct attack on traditional meat production. And that's it, right? That when you start with that adversarial position, like it gets people's backs up, right? And they start to, well, you then, right? And so overall, my position is that we all need to take a breath, right? That like it's fine. It's just another different food. And in the end, it's gonna be fine.
Oh, great comment, Anna, thanks. And I'm going to hand to Rafael Guerrero from Mexico.
Hi, hi Anna. Hi, thanks, Robin. I actually really like the presentation because I'm really keen on regulation. I worked on the government for a little bit, and I have the experience to import and export merchandises between US and Mexico. But my question is more like, is the meat product from a laboratory behaves the same as traditional meat in terms of freezing and transport?
And I think your question, Rafael, is probably around how perishable it is.
Yeah, how perishable is it? Yeah.
Probably, is it micro and oxidation, Rafael?
Yeah, no, that's a really, really great question. And honestly, I would love to get my meat science hands on a good 50 pounds of it, such that like, yeah, I'd love to answer that question for you. And honestly, I have seen no data to discuss the concept of, right? And so I have some limited experience. I'm sure a lot of you do as well, like of thinking about cultured cells, right? And when you have cells in culture, it looks very different than a muscle, right? It looks even very different than ground meat. And I have never tried to store them in a way to make them into food. Right? And so when we think about, yeah, absolutely, we can store them, but not in a way that we think about making them into food. And so I think there are major issues around microbial storage or microbial spoilage, around oxidative stability. I think there's massive questions around textural properties. And like, I think we're going to start with these cells as an ingredient, and we're going to have to do a lot to classify how does that ingredient behave. And to date, I am not aware of any publicly available information that answers that question. And I think one of the things that's really interesting about these products coming to market is they are almost exclusively being developed by private companies. And so for those of us who are really interested in it, it's really hard to get information because all of that information right now is really proprietary.
Yeah,
Thanks,
good
Anna.
question, Raphael. And I'm going to hand over to my co-host who hasn't said much yet, Dr. Minhar.
Thanks, Anna. I'll make it very quick. So from a regulation point of view, we are so far away, both in the US and in Australia, from approving anything. How did Singapore get into that so quickly?
Yeah, right. And I'm not sure that I'm not sure how many people expected that Singapore approval there in late 2020. Some of it is that the Singapore government has made it very clear that they want to be a leader in this space. They want, and we're attracting companies to them, saying, we want to be the first, we want to be a leader, we want to be at the forefront of approving these products. And so from that standpoint, I think they just really made it a priority. And it married up really nicely. What I think is interesting is, right, we're now 18 months past that initial introduction. And in Singapore, it is available on a very limited basis, but it really hasn't then branched out into other countries. I am not familiar with the regulatory environment in Singapore to tell you whether or not they kind of approach things in a way that is similar to the US, to Australia, to Europe, which is very regimented in assessing safety. And so I am not saying that the Singapore government did not make a full safety assessment. I just haven't seen it. And I'm not sure that it, right? I did some digging, and so hope maybe you've been able to find it, but like, I'm not sure that they have, there's not really been a lot of information produced.
No, there's not. And I think that's interesting because it does mean their process is a lot faster because they don't have public consultations. They're not encouraging democratic engagement with what their standards are and what they're passing. So no, we don't have a lot of information. And Singapore government itself, as we know, is investing in the development of these products. So yeah, it is interesting to compare their very sort of seemingly light touch approach to the lack of actual information. Yeah.
Mm-hmm.
Just before we finish, Min, I just wanted to, I know we've got 1 minute, and it was interesting. It was either Kieran, who's still online, or Brodie, who pointed out an article to us, I think it was in Nature Foods, where was the very first texture test done on cultivated meat. Yeah, Kieran's got his camera on now. And so we had a good read of this paper and we went, oh my God, how did this on earth get published? Because it was a product they got from a company. They didn't reveal how much meat cells were in the product. They didn't reveal the ingredients, the binders, the extenders, everything else. And they compared it to, Kieran, do you remember? What did they compare it to?
Yeah, it was raw chicken breast and processed turkey and processed chicken frankfurters.
How's that for a extremely poorly designed test of the texture of cultivated?
Well, and you're like, wow, those are like the worst textures of meat. That, right? You're like, the fact that it would be similar to that does not really make it terribly attractive to me.
No.
Right?
So we were pretty shocked by the science actually that it got published in a reasonably prestigious journal actually. Lucky it wasn't in one of your journals, Anna.
No, luckily it was not one of our journals. But I actually think it brings up this really interesting kind of space that you all now exist in that there is so much interest and so much hype that I think we're going to see publications like that honestly did not add anything to our scientific understanding, right? They were more just kind of like a woo, one-off, like, woo, that was so exciting, right? When I think what we need is serious, skilled meat scientists working in these areas. And right now, we can't obtain product.
Yeah, that's right. Yeah. So we can probably stop recording now, Min, but we'd like to really thank you, Anna, for the great presentation, but also a great discussion afterwards. I feel like we actually probably almost go on for longer, but we need to let everyone get to what they're needing to do, as well as you yourself, Anna. So really appreciate your time today. So thank you very much.
Thank you. I really appreciate the invitation, and do not hesitate to reach out. I am happy to talk about this because like I said, it's just fun to talk about.
Yeah, it is. That's right.